Namaste to all the distinguished participants of the 4th International Virtual Multidisciplinary Conference from Gurukul to Global, Paving Path to Viksit Bharat at 2047, Reconstructing Higher Educations with Indic Priorities. I would like to recall the vision set forth by the National Education Policy 2020, a landmark educational reform that emphasizes holistic, multidisciplinary, skill-based and value-driven learning. NEP 2020 clearly states that India must recover and reintroduce the rich heritage of ancient and traditional knowledge system to shape the learners of the future. This vision forms the perfect foundation for today's theme. Myself, Dr. Shalini, Assistant Professor, Department of Biotechnology and Biochemistry, Avinashlingam University, Coimbatore, Tamil Nadu. Today, I would like to speak about redefining education, Gurukul principles for holistic growth in modern world. It is an ancient education system that remains remarkably relevant even in the 21st century. While modern classrooms often prioritize marks and examinations, the Gurukul model reminds us that true educations go beyond academic and shapes the mind, body, spirit and character of every learner. At this core, the Gurukul Parampara embodies a learner-centric and value-based approach that cultivates genuine holistic growth. Today's education demands personalized learning, strong teacher-student bonds, value-based foundations, lifelong skill developments, holistic growth, and practices that nurture mental well-being and reduced stress. All these aspects perfectly aligned with the timeless essence of the Gurukul system. Gurukul inspired method enhance modern pedagogy by bringing experiential value-based learning into schools, universities and skill programs perfectly aligned with the holistic vision of the National Education Policy 2020. The principles of holistic learning mainly focused on intellectual, emotional, spiritual and physical development rooted in the Gurukul Parampara. It is now being meaningfully integrated into the modern education system through National Education Policy 2020. It mainly based on the project-based learning and experiential learning in today's school reflects its approach. For instance, students solve real-world problems like preparing a water conservation model where they research, experiment, experience and present. Similar to the analytical training in the Gurukul. Mentor-mentee system in the university helps students discuss academic stress, personal issues and career choices. Meditation and yoga periods in the school are modern adaptation of the spiritual grounding. Many universities and colleges, schools have wellness center promoting guided meditation and value-based education for the inner well-being. These activities have enabled students to experience and understand holistic learning envisioned by National Education Policy, thereby contributing to the vision of Viksit Bharat 2047. In a nutshell, Gurukul Parampara teaches that education is not just about exams. It is about shaping a balanced, thoughtful and capable human being. Bringing Gurukul principles into the modern world is not about looking back. It is about moving forward with wisdom. When experiential learning replaces rote memorization, when mentorship replaces isolation, when ethics guide innovation, and when nature once again becomes our school, we nurture individuals who are not only skilled but also grounded, responsible, and compassionate. Thank you.
Good afternoon to everyone. This is Dr. Rajkumar, Professor in Biochemistry from MGM University. Thanks for the conference. Make me as a chairperson. I would like to appreciate the program organized by Vedanta Knowledge System Private Limited and welcome to all the participants for 4th International Virtual Multidisciplinary Conference. And I received 4 papers related to the multidisciplinary activity. The first paper, which is presented on redefining education, it is Gurukul Principles for Holistic Growth in the Modern World, which is presented by Dr. K.V. Shalini, Department of Biochemistry and Biotechnology. Avinash Lingam, Institute of Home Sciences for Higher Education for Women, Coimbatore, Tamil Nadu, India. And as these people defined very well about the education system, which is started as a curricular principles, it is the ancient system of the education, as I already know, with a different kind of the pedagogy and the way of the entry into the education system from the lower level to the higher level, and which is making the experimental learning and value oriented education and a strong bond between the teacher and students. So when the Gurukul system is going to happen, there is no books and there is no pen and paper which they can do anything. But nowadays in the modern education system, we have a lot of equipment and a lot of other things. So which is making the growth easily to understand the things, and we can commit for the experimental education and where the project outcomes will make the good system. The development of this particular education system from NEP 2020, which is promoting the multidisciplinary system, which is making the students to follow different kind of the aspects as well as the critical thinking and to understand the ethical and informative system of the education. So finally, I would like to appreciate KV Shalini Madam regarding this presentation, the way she entered into the topic and concluded the things as well organized. So congratulations to the people who are contributed on this work. Thank you.
Namaste. I am Dr. Vinothini. I have worked as a private institution as an assistant professor for past 17 years. I am very pleasure to attend this conference. So I would like to present my paper. My title is Textile Study on the Textile Dye Removal by using crab cell cytogen. So crab, normally nowadays we are a lot of effluent that is present in the wastewaters. So we have to remove the wastewater by using the natural biodegradable substances like cytogen. Cytogen is a polymer that is present in the natural crab cells. So we have to collect the crab cell by using the natural biodegradable substances. So biodegradable substances is very much effective for removal of textile dye. So nowadays we are facing lot of dye removal from lot of sources. We have affected from lot of infections and biodegradable substances is present in our environment. So we have to protect our environment by using the normal polluted water is removed by using cytogen. So it is a natural substances present in the all the exoskeletons of the crabs, prawn and shrimp. So I have selected crab for this study. So I have to come to the materials. I have to selected crab cells, that is crop is collected from the local market that is available in the Shisho region is Nagapattinam, and it was properly washed and kept maintained for 24 to 48 hours, and you have to add acetic acid for removal of the that is outer layer of the skin, and that is used for the biodegradable substances. So these substances is used for the Christogens. This Christogen is used for the different parameters, that is 10%, 20% and 30 percentage and 40 percentage, and the percentage of biodegradable substances is analyzed by using decolorization process and biotoxicity assay method. And also I have to analyze HPLC for the level of, that is percentage of biodegradable ability is calculated, and I have conclude that the natural substances that is present in the seashore marine regions like shrimp, prawn and crab, all are that is outer layer of the skin is contain cristogen, that is polymer. This polymer we have to apply and remove the industrial effluent substances released from the textile industries and all industries. It was released in the waste management. So you have to remove the waste management by the application of cristogens that is collected from the biodegradable substances. So I have to conclude my topic. Thank you.
So, welcome back. And the second paper is on a study of the dye removal from the textile effluent using the crab shell chitosan, which is presented by Dr.C.Vinothini and is assisted by Miss R. Sujita. These are the assistant professors in the Department of Biotechnology, EGS Pillai College of Arts and Science, Nagapattinam, where they can express the exact waste management treatment system, which can be appreciable. But the textile effluents containing the lot of dyes, which is containing different kind of the pollutants, the way they entered into the experiment is awesome. But the thing I would like to make the science how these shells are going to be absorbing the particular chemical and what is the next step of that particular adsorbent. So how it will be changed into the biodegradable thing. So this kind of the questions arise from me. Other than that, the way of the methodology developed and the explanation is also good. And the system which we are making totally related to the aquatic animal and how it will be the pollutants can be absorbed, if so absorbed, and how it will be biodegradable. So these kind of the things we have to reconstruct. And other than that, the methodology is good and the analysis also given the good results. So where this chitosan is the chemical based absorbent, how the particular activity of decoloration will be effectively working on with corruption. So as the things are mentioned here, the results are optimum and the discussion is also well organized. And the peaks of the graph HPLC chromatogram is not seems exactly what we are expecting. So it should be reconstructive work and continue on this same work. Thank you.
Today, I am going to present my research topic on the conference From Gurukul to Global, Paving Path to Viksit Bharat Act 2047, Restructuring the Higher Education with Indic Priorities. My research topic falls under the theme Science, Engineering and Technology for Atmanirbhar Bharat. My topic is Enhancing Clindamycin Detection Optimization through Molecular Imprinted Polymer. My co-workers are Gopika and Maria Donna Paulson, and we all are guided by Dr. Danya Gambadran, Associate Professor and HOD of Department of Biotechnology, Sahardi College of Engineering and Technology, Parabathina, Thrissur. The AMR, which is the antimicrobial resistance crisis, is really threatening the global health issues. It's a major health issue across the earth because of excessive antibiotic usage leading to bacterial resistance and failure of treatment. Now, why did we choose clindamycin? Because clindamycin is the commonly prescribed lincosamide antibiotic, and it has created a lot of environmental contaminations. And its detection and rapid and the precise detection in the complex environment is very, very critical. The current detection which is done for clindamycin is through chromatography and immunoassay technique, both of which require very expensive equipments and chemicals and expensive lab resources, highly trained professionals, so which is highly expensive. Now, the revolutionary technology which we are included here is the MIP-based solution. MIP is molecularly imprinted polymer, which are synthetic polymers with molecular cavities for specific target recognition. Combined with the electrochemical sensors, they offer high sensitivity and rapid results, potential for miniaturization, that is, you can have a point of care device or on site platforms. The electrode is prepared by using screen printed carbon electrode, which has been fabricated in our lab. Then electropolymerization is a technique used for the polymerization of the monomer, which is used here is orthophenylene diamine. And the template is our particular antibiotic. We use CV at the potential range of 0 to 1 volt, 30 cycles. Then the template is removed and creating selective binding sites for our particular template in the polymer matrix. And the optimization study was done where the template monomer ratio was studied and the maximum was for the 0.5:1 ratio, and the electropolymerization cycle was done and in which we got the best and the highest in 30 cycles, and the template removal which we had done using various buffers, and we got phosphate buffer 0.1 molar PSN 0.4 for 5 minutes could effectively remove this particular template by maintaining the polymer integrity. Now we got a linearity study, or our sensors linearity range is around 50 to 1200 miomolar with an R square of 0.955 and an electric LOD at the 199.8 nanomolar. So it is very, very, very superior. Then we check for the interference study for its, that is, selectivity with similar class of drugs, that is antibiotics, and various other electrolytes or electroactive ions which is present in the blood, in which we got minimal interference. Then real sample validation was done on human serum and artificial urine, where we got the high, around 110 to 100, very high recovery rate in serum and then comparatively with urine. Maybe because of various other components which might be there, like we have the proteins or some other structures which can cause further interference. Future is you can use this into clinical diagnostic as a point of care antibiotic monitoring, then reducing antibiotic resistance through rapid detection, then even screening the antibiotic residues in the food products, detection in the water and the soil samples, pharmaceutical analysis for the quality control and the drug formulation testing. This MIP-based electrochemical sensor offers a cost-effective, sensitive, and a portable solution for on-site clindamycin detection with potential detection for expansion to other antibiotics. Thank you.
Start.
Yeah, the third paper is presented by POOJA Das and Anne-Maria Prince, Donna Paulson, and Gopika S. Nair, and Dr. Danya Gangadharan. So these are the Department of Biotechnology, College of Engineering and Technology. I think it's from Canada. So exactly I can't pronounce the name. Sahradaya College of Engineering and Technology, hopefully. So now the title of this one is Enhancing the Clindamycin Detection, Optimization through the Molecularly Imprinting Polymers. So in this particular study, they want to identify the clindamycin, which is present in the various sources. And the way of the methodology they developed is very well organized. And the abstract is also presented very well. And I would like to suggest them, so how these sensors will work in the general system. So what is the monitoring ratio? Is it a digital or optimal or visual? So which need to be explained here. And the sensor is developed very well. And the electricity and the performance wise also it is good. The results also they expressed very well organized. The chemicals, the reagents and their activity and everything is presented very nicely. And by this activity, I would like to congratulate them to continue this kind of the work for furthermore. So which is appreciable for the identification of the some of the antibiotics which are like a pollutants or which are making some kind of poisons for the food and other activities. So this work is also very good and I congratulate them.
Dr. Shilpa Deshpande Kaistha from the Department of Biotechnology at Chhatrapati Shahuji Maharaj University, Kanpur, and I would like to share a part of our work that we have done regarding microbial diversity from Mahakumbh 2025 Prayagraj. Since ancient times, our rivers are considered as holy and self-cleansing. As part of many religious ceremonies, the Mahakumbh Mela, the grandest which occurs every 144 years, occurred this year in 2025 at Prayagraj in the Triveni Sangam. Many travelers took holy dips and took water back for religious ceremonies, believing in its healing powers. Scientific inquiry over the years has revealed that this self-healing powers may be likely due to unique hydrology, the unique physicochemical characteristics of the water, and its unique microbial flora, including the presence of bacteriophages, bacterial predators, that is, viruses that lice bacteria. Even as part of our Indic knowledge, classical Sanskrit sources, Ganga Stotram, Padma and the Skand Puranas, consistently describe the river Ganga waters as Sarva Rogahara. There is also the mention of the Sukshma Krimis or the microorganisms, although there is no description of bacteriophages or viral predators or bacteria by any aim. Thus, the hypothesis of this research was to draw any connection between the classical self-purificatory claims and the modern phage ecology, integrating textual and microbiological evidence. As shown by some of the stotras here, it is very clear that there is a mention either of taking bath in the Ganga water or consuming the Ganga water, which leads to which attributes to the sanctifying power of the divine waters. This study hence was taken, as mentioned, between the times of 13 January 2025 to 26 February. This is the sampling site, and sterile water samples were brought in at pre-Kumbh and at different time points. These timelines were decided based on the festivals that occurred during this period, where massive number of people actually were present at these sites taking baths. Here is shown a brief protocol where we took the waters. In addition to doing microbial sampling, we also processed them for the double layer agar over assay, where we looked at plaques which are indicative of bacterial lysis. This is the data of the microbial and the plaque forming units in different ghats of Mahakumbh. Here you can see, and from pre-Kumbh to day 35, we have seen at different ghats the plaque forming units, which is the bacteriophage activity, and the aerobic heterotrophic count, that is CFU per ml. As can be seen, there is really not much difference, although a slight increase can be seen at certain sites in terms of the microbial activity. So here is a comparative analysis where you can see in orange the phage activity remains constant, while there is a slight increase in the microbial activity at day 5, which relatively steadies over time. Another interesting part of study that we did was that the samples that were stored with us, we took these samples out again after 320 days and again did a plaque assay for the same. Some of the pictures here show the different type of plaque assays, and what we found was that only in some of the samples we could see about 1 log difference or 1 log reduction in the number of phage. So in summary, we found that the culturable microbial load increased during peak Mahakumbh bathing days, possibly due to intense human activity or sediment disturbance. The phage bacteria dynamics rose with a short lag, indicating the presence of bacteriophage in the samples. Post-event recovery, the numbers went back to the normal. Within 10 months, we found a 1 log decline in some of the sample. The implications of public health show that this type of timeline series monitoring during mass gathering enables early risk detection, and as well as these findings support integrated rapid microbial surveillance and culturally sensitive sanitation strategies for safer events. The presence of bacteriophages during these times definitely provide evidence for a natural mechanism for cleaning of the river Ganga.
We came to the end. The paper presented in the last for me is the temporal dynamics of the total microbial and bacteriophage count from the Mahakumbh 2025 Prayagraj. It is a timeline series study with implication for the public health, which is presented by the Shilpa, Deshpande, Kaistha, and Shubham Yadav, where they are from the Department of Biotechnology, School of Licenses and Biotechnology, Chhatrapati Sahuji Maharaj University, Kanpur. And this kind of work is very interesting and which leads to the contagious diseases and make the sense how to aware of the public health and the methodology what they collected the samples and done for the different kind of the microbial activity is also awesome. So the presentation skill is also awesome. So this method and the results have a super conclusion and which is making the significant study the growth of the microbial organisms in shortest duration and how it is contaminating the total water and what kind of the precautions we must be taken for further. And this kind of the public activity like a Kumbh Mela or other activities are happening throughout the India because of we are Hindu country, so we are developing lot of Sanatana issues and all. So where the people or pilgrims should not be suffer. If this study is makes a kind of the sense to understand how we are making the hygiene to avoid some kind of the contamination diseases. So this is also a good paper. So which I just congratulate all the 4 participants on the multidisciplinary from Gurukul to different kind of the sensors they developed and different kind of clindamycin detection they done. And the madam like Vinothini madam presented very good anti-pollutant which is produced from the crab shell. It is also like waste management. This kind of the activity makes a sense and I would like to show my gratitude towards the committee. So Vedanta Knowledge System Private Limited and welcome and congratulate for all the participant who participated in the 4th International Virtual Multidisciplinary Conference. And I would like to thank all of them and thank you for giving me a kind of opportunity. So welcome and thank you all.